Cancer Center and the Center for AIDS Research. And through our sort of single cell work and working with investigators using flow or cell surface, oligo-tagged antibodies to imply cell function, we became interested in a platform that is now Beacon Discovery from Bruker, that was previously Berkeley Lights, that uses optofluidics to trap cells and enable functional assays on single cells or groups of cells. And then more recently, we heard of a platform called Cellanome, which has a sort of similar capabilities, but uses CellCages to isolate cells for functional assays. And somewhat rashly last year, I proposed that there should be a session on this after never having touched either of these instruments. A year later, I still haven't touched either of them. But that's how we came to this session on optofluidics and CellCages and different approaches for multimodal single cell discovery, and really highlighting these new platforms. We have two academic speakers and a speaker from Cellanome today. First of all, I'd like to really thank Dr. Soroush Ghaffari, who'll be speaking about Beacon Discovery. He agreed to speak at about 48 hours notice after there was a conflict with our previous speaker. And that may be the end of my introduction. There we go. Okay. And Dr. Ghaffari is at University of Texas at Arlington, where he's an assistant professor and lead immunologist at Imprint, which is the Institute of Biomanufacturing and Precision Medicine for North Texas, where he works on cancer immunotherapy and TCR mimetics. And as I say, a huge thank you for being able to hold this entire session together today. Our second academic speaker will be Dr. Simon Gregory, who's a tenured professor and director of the Brain Tumor Omics program at Duke Department of Neurosurgery and vice chair of research in the Department of Neurology and director of the Molecular Genomics Core at Duke Molecular Physiology Institute. At his institution, he's recognized as a leader in the development of single cell and spatial molecular technologies, and his primary areas of research involve understanding molecular processes involved with disease development and progression in brain tumors and Alzheimer's, and novel drug-induced white matter injury and repair in MS. We'll also be hearing from a colleague, Andy Larrea, who did his PhD at University of Miami. We sort of overlap a tiny bit, and worked through a number of genomics companies we work with, including PacBio, and he'll be introducing the Cellanome R3200 platform, which Dr. Gregory will be speaking about use cases and implementation of that platform. And I think we'll, with the same format as everyone else, we'll be taking questions for both platforms at the end. And I believe I need to remind everybody as well that unless specifically stated by investigators, photography is permitted. All right. Thank you very much. Soroush.
Good afternoon, everyone. I am Soroush Ghaffari, and I'm associated with Institute for Biotherapeutic Biomanufacturing, and today I am presenting portion of the research that my group conduct over single B-cell discovery platform on Beacon, known as Beacon. So without further ado, I would like to jump to the science. But before that, I was told I have to talk about the mission of the Imprint. What are we doing? Maybe some of you guys would be interested to start a collaboration with our team. So at Imprint, our mission is to integrate clinical insight, biological discovery, engineering, and biomanufacturing to develop a scalable precision therapeutic. This is to support by comprehensive set of capability. From antibody discovery, characterization, to process development, allowing us to make idea from early discovery all the way to the translational point. And now science. Monoclonal antibody, or known as MAB. Many of you guys know that campaign for discovery of the MABs are not simple. Sometimes involve a screening of 1,000 to a million hits in a hope to find few that they are matter. However, we were thinking about what could be if we are focusing on those immunological compartment that has important role in this case. And in our case, it's going to be B cells. So monoclonal antibody have become basically a cornerstone of modern therapeutic, especially in oncology, autoimmune disease, where they can precisely modulate immune pathway or directly target disease cell. However, the field rapidly evolved beyond traditional naked antibodies. We now see rise for engineered formats of the antibody, like bispecific, which simultaneously engaging to basically target at the same time, bringing in most cases immune cell in the proximity of the tumor cell and activating the immune cell to directly kill the tumor cell. Antibody drug conjugate is another horizon in the field of immune engineering, which basically targeting the antibody that carrying payload of the drug directly to the tumor. With that said, we're reducing systemic toxicity. At the same time, we take advantage of antibody specificity and targeting a tumor cell directly. Additionally, other method taking advantage of basically modality of the way that antibody recognizes target, develop and take advantage of the cell therapy, and those the way that antibody recognizes target is called CAR-T therapy, and basically create another pioneer in the field of immune modulation and immune engineering. So overall, antibody should no longer be viewed as mature or static technology. They are a rapidly evolving platform with expanding functionality and therapeutic reach. Like conventional antibody, TCR mimic antibody is another form of the antibody that my group are having interest in developing them. And like many other antibody, is recognize a protein. The only difference is the protein that this form of antibody recognize are peptide major histocompatibility complexes that express outside of the cell. One unique fact about this target protein is that indirectly basically presenting what's going on inside the cell. If you have internal protein, it's getting processed and being presented via peptide MHC on the surface of the tumor, and TCR mimic antibody is acting like T cell receptor with the specificity of the B cell receptor, and technically specifically bind to the target and can do what it's supposed to do. So like many other antibody discovery campaign, TCR mimic has its own challenges. This brings us to the central challenge of the field. While TCR antibody allow target basically be recognized via peptide MHC biology, some of the challenges are as listed below. First, peptide MHC complex presented on the cell are very few copy number. In some cases from 10 to 100 copy, which is make it very challenging. Second, peptide repertoire is highly similar, meaning antibody must distinguish very subtle of the difference, sometimes as small as single amino acid. And then third, which basically this causing significant specificity challenge, where avoiding cross-reactivity is critical for us. Finally, B cell capable of producing such high specific antibodies are extremely rare, making them difficult to isolate using conventional approach. So overall, antibody we need exist, but they are rare and challenging to find. This is exactly what we propose in collaboration with Berkeley, formerly Berkeley Light, now Beacon Discovery, part of the Bruker platform, to overcome this challenge. I guess, okay. So historically, monoclonal antibody discovery campaign use some method. The most famous one is a hybridoma technology. This approach has clear strength. It preserves natural pairing of heavy chain and light chain and allow functional screening of the secreted antibody. However, when we think about these challenges we just discussed for TCRM discovery, hybridoma start falling short. The process is relatively slow and often taking week to month of the sample prep and then till you reach the final clone of the antibody. It also low throughput, meaning you can only sample a small fraction of the total B cell repertoire. And most importantly, it is not well suited for capturing extremely rare clone for TCR mimicking antibody. So while hybridoma have been functional and fundamental in the field of antibody discovery, is not ideal for my application, which is TCR mimicking antibody development. So if we step back, what we see is a fundamental trade-off in the antibody discovery. On one side, we have hybridoma technology approach that preserve natural biology, native heavy chain, light chain pairing basically, and functional context, but lack scalability. On the other hand, we have technologies such as display of phage and yeast, which provide us enormous power of the selection, which is critical to finding of such rare binder. But by doing so, we lose some of the biological context, particularly natural pairing and physiological relevance. So the field essentially had get stuck between two extremes. We gain a scale at the cost of biology or vice versa. What should we do to overcome this barrier? So coming back to the question, can we achieve both scale and biology? My answer would be yes. Single B cell driven biology offer a way forward for antibody discovery campaign. This approach preserves a key advantage of traditional method, including native heavy chain and light chain pairing, and the benefit of in vivo immune selection. At the same time, it enables functional screening at the single cell level, allowing us to interrogate antibody secretion specifically much more efficiently. Importantly, this can be done rapid and in a scalable way, overcoming many limitations that hybridoma approach had. In our work, we leverage this technology, which is known, as I said, Beacon platform, to overcome this boundary. Here, I put the timeline comparison. The platform that we use is called, again, Beacon Discovery, and as you can see here, we can effectively reduce the timeline that is necessary for a screening of each campaign from 2 to 3 months to 2 to 3 weeks. 2 to 3 weeks is from A to Z involving a characterization. Not everything gonna be on the Beacon Discovery. So here in this slide, we summarize end-to-end workflow that you'll see when you're planning to use Beacon Discovery platform for antibody characterization. We start with either immunized animal or donor-derived B cell, where individual cell capture by proprietary chip, after isolation by proprietary chip called nanochip. And then we then perform real-time secretion. We pen the cell into each of those tiny micropen, which in the next video I'm going to show you a video of one of the experiments we conduct. Then we run the assay, real-time assay, and we monitor amount of antibody that in real time secreted by each B cell and how much of them interact with our antigen of interest, and if some of those pen that are reacted to our antigens, do they present a cross-reactivity to the control antigens or not? Then after we finalize the assay, we find out which clone are the one that we are interested. We tell the signal to the machine that start unpenning them, isolating those single cell B cell, and send it for downstream characterization, which involve the Sanger sequencing to identify the VLVH and then production of them and down, so-called. After one of the advantages that I like to mention here about the team at Bruker is their customer service is very great. I keep complaining that this process is hard, and I'm coming from academic background, that dealing with the undergraduate student, teaching them working clean and amplify complementary DNA and then amplify VL and VH, overlapping them, it was a big challenge for me. So I was asking them, is there a way for you guys to facilitate this process for me at least? So after a month back and forth, they release a platform that we can synthesize a cDNA within a chip, which is basically you make your reagent, give it to the machine gonna do the job for you, which is a big plus for my team. Here is the video that I would like to show you guys. It shows how those single cell is penned into each nanopen. Then we are coming to the point that performing the assay. In real life, we can see that antibody start produced because of the miniaturized structure that those nanopen has. Then after antibody start producing, as accumulate, it start coming out of the nanopen. And then we are touching the, basically loading the chip with a series of antigens, control antigens and relative antigens. And upon interaction that we see between the antigens and antibody, we can see that fluorescent platform happen, and we call it blooming. And blooming happen at the opening of each nanopen. Those are the ones that if ended up to be not cross-react, we can unpen them and select them. And as you would like, you can make the assay more complex. You can involve them with more fluorescent and screen for 2 or 3 antigens at once. Leveraging two basically key innovation, specifically for the antibody discovery, we created basically several products in collaboration with Bruker system that involve basically all of them loading those chips. At the beginning, when we start working with Bruker only offer us for B-cell nanopen that contain 10,000 chip, 10,000 tiny micro basically chip, and then upon our request they increase it, they make 14,000, and now they have 20,000. When you are screening a campaign for the B cell, it's important to capture as much as possible within the first round. And this is a workflow in basically brief. It starts from the loading a B cell into the pen, performing the assay, capturing the bloom, and isolating those single B cells. What distinguishes basically our method? To summarize, Beacon platform bring together several key advantages. It preserves native heavy and light chain pairing and operate at true single cell resolution. Importantly, is enable direct functional screening rather relying on indirect readouts. This allows for much faster discovery timeline on the order of day rather than week or months. And ultimately, these enrich antibodies that are not only specific, but also biologically relevant. So overall, this approach combines biological fidelity with a scalable discovery. Now, where Beacon fits and some important consideration. While Beacon platform is a powerful tool, it's important to understand where it fits within a broader discovery pipeline. First, it's not designed for detailed kinetic characterization, so follow-up biophysical assays are still required. Second, there are a trade-off between screening depth and assay complexity. As you increase assay parameter, throughput can decrease. And finally, candidate identify on Beacon still require downstream characterization. So overall, Beacon serves as a highly effective front-end discovery tool that basically integrate with the downstream application workflow that our team developed. Our team basically developed a label-free technology that works, is truly 96 well plate discovery called UltraMap Pro, and work exactly like ELISA plate reader, but with guided principle called guided resonance mode. which is basically, in simple word, is worked by change in the surface density. This assay can be used for protein-protein interaction. Also, it can be used for the cell morphology change. If you are performing some killing assay that the morphology of the cell change, we can capture it with this platform. To put this into context of the beacon serve as a powerful front-end discovery tool, but is not designed for detailed biophysical characterization. To address this, we integrate our label-free platform for downstream characterization. This allows us to measure binding kinetic and affinity of each antibody, which giving us a quantitative understanding of how strongly they interact with the target. In addition, we can perform epitope binning experiment. What does it mean by epitope binning? If you might, in some campaign that you screen your B cell population, you might have more than one antibody against the same clone. You would like to know, are they binding to the same epitope or not? And if they are binding, which one of them bind stronger? Do they blocking each other's interaction or not? And with that said, also you can determine if this antibody are cross-react at the end. So together, this workflow allows us to move forward from single cell functional discovery to precise biophysical characterization of each antibody candidate. Here I show an example of some of the candidates that my team generate by Beacon and characterize using label-free platform. On the left panel, you can see a top clone that this is against the peptide HLA-E that we made, and we licensed it to Boehringer Ingelheim. Now is part of their portfolio and can serve as a checkpoint blocker. On the right panel, on top, you see an antibody that starts showing some cross-reactivity to the other peptide variation that has a single amino acid difference. And at the bottom, you see one of the other clone that we find is recognized two of the isotype with the same intensity. So with that said, it shows us real-time characterization and high throughput. When you compare this technology with the other technologies such as SPR or Octet, for those of you guys who work with that, although they call it 96 well, but in reality it's not 96 well, because some of the well is used for the dipping up and basically regeneration of the surface of the plate. Looking ahead, we see several directions where this approach can expand. From platform perspective, integrating rapid expression system and sequencing pipeline will further accelerate the transition from discovery to characterization. On the side of application side, this enable development of TCRM-based therapeutic as well as more complex format such as bispecific antibody. And scientifically, our focus while using this is still understanding how HLA biology modulate immune system. So overall, the goal is to seamlessly moving from discovery toward therapeutic translation. The key takeaway here is basically Beacon Discovery platform give us a high throughput screening platform that preserve native heavy chain and light chain pairing. In particular, is important for challenging target discoveries such as peptide MHC complexes. And when combined with downstream characterization, this creates a fully integrated workflow from discovery to validation. So overall, the feature of antibody discovery is cell-based, single cell, and integrated. And this is the framework we use to identify our antibody and at our institute. Here I would like to thank the team that I have, our sponsor, Resonance Center Boehringer Ingelheim, TCU School of Medicine, Cook Children's Hospital, and my team, which is this work was not possible without their support. Here I have the basically contact information for my team, and if you are interested to utilize Beacon Discovery in your future research, our team actively looking for collaboration. feel free to scan the QR code and get in touch with us. We would like to have a meeting with you and talk about science and see how can we help your project.
Take questions for everything at the end.
Okay, great. So I'm Andy Larea from Cellanome, and I am pretty much the intro act for Simon, who will talk a lot about how the technology is actually used. And so I'm going to introduce how it works. And so just some kind of basic background. So thanks to Sean for letting me jump in here for a couple of minutes. Cellanome is a kind of full product workflow that we've developed. So this is the instrument on the left, the consumables, as well as the software, and that software has a cloud component to help kind of bring together the real-time live cell imaging that we're looking at, as well as the sequencing on the back end. So how is it that we do what we do? We're isolating single cells, and the way that we do that is we have some kind of cells in culture. These can be primary, these can be cell lines. These get mixed on the instrument with a gel monomer. This is loaded into a single lane of an 8-lane flow cell. So we have 8 of these. Each lane is an independent experiment, an independent reaction. The software will look at the cells and then hit the gel monomer around individual cells or clusters of cells with light of a very particular wavelength. That will cause the gel to polymerize. And so what we then create is a semi-permeable gel matrix around single cells or small groups of cells. So we can look at reactions in there. Once these cell cage enclosures are created, we flush it with fresh media. That gets rid of any cells that are not present in a cage. And now we have these cells fixed in there. One of the key points is kind of down in the bottom right-hand corner there. The gel is just kind of almost like a chain link fence around the cell. So you can pass media and small molecules as well as antibodies through there. So anything up to about the size of 350 kilodalton can flow through that semi-permeable membrane around the cell. And we can work with either suspension or adherent cells. So just to kind of show what the workflow looks like, here you have an image which you may or may not be able to see, but believe me, there are cells. This is a single field of view. The software will recognize where the individual cells are, and then very quickly create a mask and flash it with light to generate these cell cage enclosures. Once we've done that, we wash away any of the cells that are not present within CCEs, and we, in this case, allow the cells to adhere. So the cells can stay on the flow cell for hours, days, weeks, depending on the exact experiment that you're trying to do. In this case, if they're adherent cells, they'll grow within the confines. We can then pass different cell surface markers and stain the surface to be able to visualize what's happening. If these are live dyes that can get through the membrane, we can also look inside of the cell. We have software developed for looking at cell morphology, looking at how certain perturbations, small molecules, genetic perturbations can impact the shape of the cell, what the cell is doing, and really looking at kind of live cell assays. So anything that you can do in a well, you can do in this cell cage enclosure. We really turn instead of putting a cell into a well, we build a well around these individual cells. And then the final point of this, if the goal of this is transcriptomic, the flow cell is labeled it has an array of barcodes present, and then at the end of the experiment, we can lyse the cell within the cell cage, capture the RNA, and then do downstream sequencing assays. And so then now here we're able to kind of unambiguously connect the cell, the perturbation, and the transcriptomic profile of those cells. What this lets us do now is we have kind of a toolkit for cell biology by looking at cells that are captured. How is it that we do this kind of longitudinal assay, and then at the end, the transcriptomic readout? And so this is the kind of the fundamental way that the technology works. It can be applied across a large number of different applications to look in cases where you want that ability to perturb, whether genetically or chemically, to do that perturbation and monitor it. And again, everything I've shown is single cell, but this could be done in a case where you have different cell types. So you're looking at cell-cell interactions, you're activating one cell and looking at the impact of the other. You can think of each one of these little cages as an independent experiment. So that is the basics of the technology. I will now hand it off to Simon, who will explain how he's using that in his lab.
Okay, thanks, Andy. So, so we've actually had the platform for about 12 months now. As with any case of getting early access to technology, you have the pleasure of evolving experiments as well as experience with the development of the technology. And I just want to say from the outset, the Cellanome guys have been phenomenal to work with. So as Andy said, you can use non-adherent, adherent, you can use mixed culture, and you can allow cells to grow on the surface of the media. And so what I thought I'd do is the first few slides are just going to have a little bit more detail on top of what we've found with respect to the platform itself. And then I'm going to go into some slides about actually how you would use it in the context of a core, and then how you can use it in the context with other cores at your institution. And so what happens is that you add around 100 microliters to the reservoir at one end of the flow cell, and the fluids come out the other end, which is called the chimney. And then, as we saw, you have bright field imaging that allows you to identify where the cells are within the flow cell. But you also have fluorescent. You have 3 different wavelength filters that you're able to capture fluorescence from the cells. You capture what you want, so you can capture positively or negatively against that fluorescence. And then, as Andy said, you use 3D printing to actually capture the cage. And then, non-adherent would be in suspension, adherent would be adhering to the surface of the slide. The surface, the distance between the 2 solid surfaces is 75 micron. So that's sort of important if you want to get around to doing things like growing organoids. And it sort of places you in the field of doing 2D organoids instead of 3D organoids. But it's something that we're currently exploring. And then, as Andy said, you can flush through antibodies, drugs up to a certain size. And you can also, at the stage where you're integrating the proprietary matrix, you can actually include beads as well. And so those beads can be reporter beads, so if they're conjugated with appropriate reporters, you're collecting cytokines, chemokines, etc. that are expressed within the cage. There's a stochastic distribution of those beads, so if you capture a bead in the cage with the cell, then you're able to detect what the cells are actually expressing. So this is one of the PIs we work with. On the left, Mike Brown is a young investigator in the Brain Tumor Center, so he's interested in oncolytic viruses and how they can be used to treat glioblastoma, which is a very aggressive brain tumor. This is what the flow cell looks like, so I thought I'd give you a picture of the actual size, next to someone's hand to give you an idea. In this experiment, we've got the reservoir at the top, chimney at the bottom. We've used the 4 different lanes in the middle. The machine itself, and so within the machine you've got buffer plates, user plates, you've got a chiller tray, you've got tip racks, and then the flow chamber is on the right where the microfluidics occurs. To give you sort of an idea of the size of the machine. It's about 3 foot square, or 2.5 to 3 foot deep, and then the machine over here is our Quanterix machine, which is, sits on the floor. So it's about half the width, half the height of a Quanterix platform. And it doesn't need to sit on an air stable, it just needs to sit on a very solid surface. Hopefully this will work. This is a video of the capture of the cells themselves. So, each of these different features is a different cell, which is recognized by the bright field. And so you'll have superimposition of what the rubric is for recognizing the cell, and it's literally a bright field activation or cross-linking of the matrix that captures those cells within the matrix itself. Okay. So important for us was variability with what we could capture within the machine. So, like I said, you can capture by fluorescence, or you can capture in the absence of fluorescence. But importantly, the cage can be altered in size. So our early cages are around 30 to 40 microns, which is too small. We have a standard now around 60 to 70 microns. So when you trypsinize cells, if they're adherent, they're going to shrink down. You mix them with the matrix, flow them down the flow cell, and then usually around 60 to 70 microns is fine. Having said that, you can have much, much larger cages. So you can have 600 micron cages where you can grow, 2D organoids or neuroblasts, allow them to multiply within the larger cage. And then interestingly, you can actually include windows within the cage. So you could have an organoid, you could co-culture with other cells and allow those cells to actually develop processes that engage through the window, through the multicellular feature within that cage. And so we've had been thinking about a thought experiment where we grow a GBM organoid, we co-culture with neurons, you allow the neurons to extend their processes and synapse with the organoid. And then, as Andy said, you're able to capture the expression of features within the surface. Also important for us was trying to understand sort of another dimension of single cell profiling. So not just looking at the expression, whether it's RNA expression or protein expression or expression in the context of some sort of spatial modality. Here we're actually using the morphology of the cell, and because you can maintain the cells alive over a period of time, you can look at the morphology of the cells. You then use cell segmentation, which allows you to develop a Seurat object around that cell segmentation. So if it's a Seurat object, you can develop a UMAP around it. And if you have a UMAP, you can combine the UMAP from the morphology together with the sequence data that you're also going to derive from the cells within the cage. And so you can start correlating morphology or response to environmental stressor with the expression that's going on within the cage itself. So this is another slide from Cellanome, which is sort of interesting. It's the use of olmutinib, which is a monoclonal antibody to treat lung cancer. So on the x-axis, you've got duration of exposure, and then the y-axis, you've got phenotypic change of the cells with concentration of the drug. So you're able to do these sort of drug assays within cells that you have caged, particular cells that are important to the phenotype. Okay, so how do we use it? So invariably we work with PIs who have particular interest in the capabilities of the platform. So we'll start with PI investigators. So I'm one of those PIs. So as Sean said, I do Alzheimer's, multiple sclerosis, and brain tumor research. So that's the benefit of being the core director, is you get to play with new toys first. But we've expanded that across Duke to look for forward-thinking investigators who can see the benefit of the technology, which is something we all have to do as core directors. We then reached out to a tissue culture core because the cells need to be alive if you're going to obviously reap the benefits of the platform. And so we are now talking with the TC core. If the investigator themselves don't have the capabilities of tissue culture, the machine sits within my core, so the molecular genomics core at Duke. We then take the libraries, if we generate them, for the RNA profiling of the cages to the sequencing core. And then we engage a bioinformatics core. As it happens, we have a bioinformatics core within the MGC, but there's also the opportunity, obviously, if you have a distinct core, to take the sequence data from the CCEs and then work with the bioinformaticians to correlate things like morphology or exposure with RNA profiles. But what invariably happens is that we have a PI who knows how their cells operate. They know how their cells are cultured, how they respond, what it is that they're looking for. We'll work directly with them, and then we'll work with the bioinformaticians within the group. And then it's just an iterative process. So how's the pilot 1 go, how's pilot 2 go, etc. So things that you need to think about are what cell type's going to work. So if you can culture the cells in tissue culture, they should be able to work on the platform. We haven't found a cell that hasn't yet. It's just a matter of sort of tightening the conditions for the growth. And then you have to say, is it adherent or non-adherent? So the surface allows you to use laminin or fibronectin. Think about cell size. So cell size going in, but also what is the cell size of the cells once they start growing. Hopefully they've previously been grown, so you know the TC conditions. Therefore, you need to know the media. Now, it's different because you're working in a very small volume. So you're working with 45 microliters in that lane as opposed to in a tissue cultured flask. And so the kinetics of cellular growth is going to be different. And so you then have to think about how often you're going to change the media for the cells, because you're effectively doing the liquid handling and the imaging on the platform, and the flow cell lives in an incubator hood at all the other times. So think about treatment. Do you want to treat the cells or not? If you are, how often do you want to treat them? And then obviously think about the concentration of treatment. What's the concentration of cells? So we've found sort of a sweet spot is around 1 million to 2 million cells per mil, and remembering that you're putting 100 microliters down the flow lane. Staining works really well. Antibodies pass through quite easily and bind or stain really well. Think about the wavelengths. You've got 3 different filters, but also important to do some sort of test before you do the stain to make sure that the antibody is going to stain the proteins that you're interested in. And then, of course, do you want to look at the RNA, which is the bar-coded version of the flow cell? But we also try to incorporate an annexin 5 stain just to look at, how alive or not alive if the cells are dead before doing the experiment. And then obviously sequencing depth. So also for this audience, I threw in another slide, so looking at project initiation. So how long does it take you to do all these experiments? Labor takes a bit over 6 hours, but that's sort of in the setup of the experiment. Cellanome have done a lot of automation for the setup of the lab protocol. And so that will populate based on sort of queued questions at the start, so that labor is going to be decreased. But then you have project management. For the machine, you've got your startup and wash and priming, counting cells, prep reagents, flow sterilization, and then the cage formation themselves. And then you can either do manual hands-on time with the machine, so changing or adding the cells, changing the media, or you can actually use the instrument to do that yourself. And then you have hands-on time for library prep for mRNA, for CRISPR-seq, and then for mRNA-seq. So that's sort of the setup of the platform, what its capabilities are to date. What I thought I'd then transition to is to take you through the projects that we've applied it to. So we've looked at CRISPR perturbation of neurons to look at morphological change of these CRISPR perturbations. Directing the brain tumor omics program, we've done a lot of brain tumor related studies, so glioma and co-culturing glioma cells from a cell line with cancer-associated fibroblasts, combining these GBM cell lines, looking at CAR T therapy or virotherapy, looking at radiation therapy agonists in a GBM cell line. We've also branched out into adipocyte differentiation, We've been able to get protoplasts to grow on the machine, which is kind of cool, and then neural stem cell differentiation. So I'm just going to spend the rest of the time giving you a couple, 2 to 3 slides of a couple of these projects, just to get you thinking about how it might be applicable to the core environment that you're in. So this is a collaboration that I have with Eric Benner. So Eric is a neonatologist at Duke, and so he's interested in mechanisms associated with cerebral palsy, neural stem cell differentiation, etc. And so what we want to do is get back to basics and try and understand the processes associated with neural stem cell differentiation. So what we're doing is deriving neural stem cells from a transgenic line derived from the subventricular zone. So here, you know, in the SVZ, you have these quiescent neural stem cells, then you have amplifying neural precursor cells, and then you have migrating neuroblasts. And so we're deriving our neural stem cells from the region of the mouse. This is a couple of screenshots from the workflow from the cloud-driven analysis software that Andy was talking about. We had 4 different lanes that we looked at, and this is just highlighting one of those individual lanes. We're fairly conservative, so we caged around a bit over 2,000 cells, and these are what the cells look like within the cage. So what is it that we're trying to look at? Take these neural stem cells and then try and understand how from a single cell you can get either oligodendroglial lineages, astrocytic lineages, or neuronal lineages, and we want to map the decision points that give rise to these different cell types. And so this is the same cage on the left and then the same cage on the right. So we have TD tomato labeled neural stem cells from the transgenic mice. So those 2 cages after 1 day under bright field, you can see that they're changing their morphology already. Day 3. Day 5, we did another stain to make sure they were still retaining those neural stem cell markers. And then on day 7, we were using 2 different fluorescent markers to define astrocytic lineage or oligodendrocytic lineage on the left and the right. So we're starting to see morphological change and differentiation along that the pathway that's on the left there. And then we had some cages that were neuroblasts were having a whale of a time in the cage by themselves. And so the next experiment we're going to do is to repeat that. So resect the neural stem cells, culture them for a little bit, split them, grow them a little bit more, and then the 1 million cell transfer that I talked about, but we're going to stage it. So we'll have stopping points in different lanes, and at each of those stopping points, we're going to look at the expression profiles. So not only bright field imaging, but also fluorescent imaging to make sure that we're seeing what that differentiation of the glial lineage will be. And then once you have all that data, you can then combine it and look at trajectory analysis like you would with traditional single cell like we do with 10x. So that's one experiment. The GBM and the CAF line, so we're really trying to understand how, what's the impact of these cancer-associated fibroblasts on glioblastoma cell lines, so U87, which is a workhorse of GBM work. So this is at day 0. Again, it's a fluorescently labeled GBM at day 1, 2, and 3. You can see it has grown and then divided. You can, within the software that's been developed for the Cellanome platform, it's sort of like a FlowJo type approach, but here you're actually changing on both. You can use it for morphology or eccentricity, so the shape, or the fluorescence that's present within the cages themselves. They've done a really cool thing of, so if you click on one of these dots, so each dot's a cage, so it could be multiple cells within a cage, it'll actually take you to the exact cage on the flow cell. Okay, so it's not wondering what this weird little cell off to the right is or cluster of cells. It'll take you to a picture of the cells, which is pretty amazing. And then, as Andy said, you know, you have barcoded oligos. They're going to give you an expression profile. So these are UMaps of expression now. So each dot is a cage or RNA from a cage, and the UMap is based on RNA expression. So you can see differences between CAFs and U87s, but interestingly, the differences between U87s with CAF media and the U87s by themselves. And you can do things like feature plot, so you know, look for the expression of particularly canonical genes. And then in this case, we're showing CAFs, you know, with themselves have extracellular matrix and remodeling gene expression within them. Do your volcano plots, so comparing, in this case, it's comparing U87s U87s plus the conditioned media. You know, your heat maps, and then you can draw conclusions about the impact of the conditioned media on permissive growth and endoplasmic reticulum stress. Co-culturing showed proliferation and stress response. And then the next step in that experiment is to take GBM cells from a patient rather than a very tired U87 line. So sticking with brain tumors again, this is work with Zach Reitman and Sheba Jacobs. So Zach is a neuro-oncologist at Duke, and Sheba is a trainee within his group. So again, looking at U87s, this time we're going to use LN18 cell lines, which is an epithelial-like GBM line. But what we're interested in is the impact on radiation therapy on cellular morphology. So the point of radiation therapy is it introduces double-stranded breaks in the DNA. So once the cell has established those, is forced to have those double-stranded breaks, the endogenous machinery within the body recognizes a cell with double-stranded breaks and then attacks these cancer cells, hopefully, or pretty much all the cells that have the breaks, and then destroys them. However, we have within our biology or biological systems two mechanisms for recovering those double-stranded breaks endogenously. So one of them is non-homologous end joining, and the other one is homologous recombination. So if that happens within the cancer cells, the cells survive and then keep on proliferating. So what Zach and Sheba are interested in is radio-sensitizing these GBM cells by using an ATM and a DNA PK inhibitor, and then looking at the effects of plus-minus radiation plus the inhibitor itself. And from their bulk RNA work, what they identified was the expression or elevated expression of CCR4, which actually plays an important role in T-cell recruitment and activation. So this is what an individual LN18 cell looks like after 0 hours, 24, 48, and 72. So they're obviously growing and proliferating. So what happens with those cells in the context of treatment? So you have the ATM and DNA PK inhibitor. What you find is that it actually decreases the amount of proliferation, but then also finds a correlative expression of this CCR4, so what we think is an important modulator going forward. Because you have morphology, you can, and you're actually counting the number of cells within a cage, you can see the proportion of cells in the cage, so whether they're proliferating or non-proliferating based on the presence or absence of the inhibitor plus radiation. You can look at individual expression, but not of the RNA in this case. It's the fluorescence of the binding fluorescently labeled antibody. And then you can look at morphology. So Andy mentioned eccentricity, so you can look at the shape and size of cells, so you can actually sort on a sort of, again, a FlowJo type plot of the size of the cells. And so what's been really cool about this study, it shows, that it's upregulation of this CCR4 plus the radiation. So this is in a paper that is being submitted, which includes the Cellanome data, and then suggests the possibility of using CCR4 as a potential therapy, and it's actually being used currently in immunotherapy trials, so it may be a really interesting repurpose of the drug. And then finally, we didn't think this would work, but it did. We were actually working with Kelsey Reed and Bas Bargman from Virginia Tech. So they're interested in working on plants, and so they're trying to understand what gene regulation is involved with allowing cells to enter cell cycle and proliferating or de-differentiating. And so what they did was took a Arabidopsis, which is the workhorse of the plant world, isolated protoplasts, so effectively plant cells without a cell wall, and then they're interested in seeing with these calluses which go on to develop the root or the stem, etc. And so what they did was isolated these protoplasts and then hot-footed it down the 80, so they were about 3 hours away. And then on the right, you see these protoplasts with the fluorescent labeling on the Cellanome platform. So we carried out the experiment. We didn't think it worked tremendously well, but we analyzed the sequence data anyway. So what we did is took the top 10 markers, generate the heat map, and then VJ, who's the lead bioinformatician within the team, looked for canonical markers of cell type, and then we're able to establish from just these protoplasts, they're starting to develop into epidermis, mesophyll, vascular, root cap, etc., which is very cool considering we didn't think it was great data. So imagine what it's like if it works really well. So they're going to go back and look at different lysis and different treatment conditions, and then we're going to allow them to grow a bit further and then see what we find as the genes that are associating with expression. So just finally, what are we doing? So with the platform, so it's, as I've shown you, it can capture cell activity. You can do cell-cell interaction analysis. One of the things I didn't mention is we're looking at CAR-T co-culturing CAR-Ts with with the target cells to see the efficacy of those CAR-T cells. You can look at cell morphology by cell segmentation, and you can do cell screening for particular assays. Talked about the neural stem cell differentiation, the CAR-T efficacy, Mike Brown's work, and the STING agonist, Zach's work on sensitization to radiation therapy. We're also looking at pre-adipocyte combination with drugs to try and drive the differentiation of adipocytes. And interestingly, one of my students noticed that the cDNA that you get off the machine is actually quite long. It's around 800 to 900 base pairs, which means you can start using it for long read, which then means you can generate isoform profiles and then correlate very granular RNA profiles with morphological change, so going beyond just your standard gene expression. And then I talked about the CRISPR-mediated neuronal perturbations. These are the guys, so Emily, Allen, Stephanie, and Karen is actually the core manager of the MGC. VJ leads the bioinformatics. And then please feel free to look at that QR code. It'll take you straight to the MGC site. We would welcome questions, interactions. The whole deck was set up so you could take photos, which is good to see people taking photos for a change. And yeah, just follow up with me if you have any questions on top of that. Okay, thank you.
Thanks very much. Thank you, Simon and Soroush. I'm not entirely sure how questions are going to work, and I apologize for the lighting. We should have fixed that. I believe there are microphones if anyone has any questions, or you could just yell. I don't know if you want to perhaps stay at the front just for a minute or two, Simon and Soroush.
Yep.
Yeah, perhaps if you sit down, I'll try this
So if I'm distracted, it's because Duke's playing Yukon at the moment.
They're
Sorry.
losing, right? Yeah. So there was a couple of papers in Science a month or two ago with these, again, this type of semi-permeable fixed cages, but they were in solution, didn't need any type of architecture, just or minimal instrumentation. I don't know if you saw those papers or if you did, and you can compare how your methods are similar or dissimilar to those.
Yeah, so the big... So most of those... Is this actually working?
Yes.
Okay, great. So most of those are in solution, and so you can't get kind of longitudinal look images of those cells over time. So they are single cell capture. They does keep the cells alive, but it is difficult to connect the individual cell to any kind of biological change within that cell, and then to the single cell. So that's kind of where we see the primary difference, and those are also working, as far as I saw, exclusively within suspension cells. So the ability to also work with adherent cells, I think, is a difference. So.
Is this on? Okay. I have sort of a similar question about like the logistics and workflow of like what is on the instrument versus how much babysitting is needed as far as keeping sort of the experiment going with the cells once they're on the flow cell, and, you know, media and like no one's talking about like when this crashes or like all your cells die. So like what are the things to be kind of cautious about? Or
So we obviously didn't launch into the expensive RNA profiling experiment to start off with. We got flow cells because you can view them under a normal confocal microscope. You don't need the scope on the machine to be able to provide the images. So all the caging steps, yes. And then if you keep an eye on how the cells are looking, and if you know your cells, you know when they start looking ill. And it also, by doing that, benefits you if you just get an idea about when they need media changes. So we usually start with what they normally get in the TC dish, and then alter from there, and then, you know, change the frequency usually, because it is such a small volume, and you can imagine if you've got, you know, up to 10,000 cells caged per, you know, 45 microliters down that length, there could be quite a lot of waste coming out of the cells, so you want to keep them happy, so we change them reasonably frequently, but we will always check them under the confocal just to see what they're doing, because we were really surprised that neural stem cells changed so quickly and starting to fate based on their morphology really quickly. And so that then changed the speed at which we're going to do our experiment, because originally we're going to do 2 weeks alive and then have stopping points along there, but we pretty quickly realized that we could probably get the data that we need to see the fate mapping of those cells within a week or 9 days. So I think it's just getting used to how the cells behave in a different environment and adjusting your media changes and the concentration of the exposures that they're going to have as well.
okay, thanks, that helps. And then you just also kind of addressed my second question, which was, we're looking at these really beautiful pictures, and it's so cool that you can kind of stain and track the individual cells, but I didn't have a sense of, yeah, how many cells are kind of like in each lane or like within the cage, and like what does that work towards for, like, if you are going to go ahead with the RNA sequencing, and kind of what do those next steps look like?
So I don't know how many features in a lane, you know, the barcoded features, I don't know. But there's way more barcoded features to generate expression profiles from than there are caged cells. One of the things that we learned was our first neural stem cell experiment looked fantastic. And then we thought, okay, next time we'll wash away all these interstitial cells because we don't need to look at them. And then the second experiment shows that those interstitial cells are really good to have around because they're secreting stuff that keeps what we thought was the neural stem cells happy. And it is really interesting because you have a physical property where the cells are caged. Cells from outside the cage will migrate towards the cages and then encompass them, which is sort of weird. We weren't expecting that. And so the third experiment, which I showed you, we're not going to wash away those interstitial cells, but because you have the barcodes that cover the surface besides the cage, you're capturing the expression of them as well. And then you go through the usual thing of, you know, stratifying them based on known canonical markers of those cell types and things like that for cells at a single cage or, you know, outside the cages.
Okay, that's good. I was thinking that you were only able to capture what was in the cage, but you're able to get anything that's on the flow cell because it's all barcoded.
Yeah, and because
okay.
the cages are geo-mapped, if you like, you know which cage corresponds to which barcode.
Yeah.
So you can figure out, okay, these features are inside the cage or outside, and then what they're correlating to with respect to, even if you didn't do the cell segmentation.
Thank you.
One more question. You mentioned the observation about the length of cDNAs. What in the method do you attribute that?
Just because it's oligo-DT prime, so it's 3' primed.
Okay.
Yeah, so it's not probe-based, and I think we're pretty excited because I don't think changes in morphology are going to be explained by a gene. I think it's going to be explained by an isoform of a gene, which we usually ignore, honestly. And so I know that these guys who've run the experiment, we tried to figure out how to use PacBio's Connects to take the libraries that were produced by the flow cell, but it's just too complicated. So these guys just stuck it on an ONT and seems to work fine.
Actually, I've got a question. So, Soroush, I heard you mention that on Beacon now there is an in situ cDNA workflow, which I wasn't aware of. So, does that generate a cDNA library for a pen? And if it does, how do you get that out?
With the same method that we are gonna use the basically charge force, I guess.
Oh, that was on.
Is it? Oh, yeah. With the same method that we use the charge force to unpen the single cell, we can unpen those cDNA into the 96 well plate and utilize them for the sequencing.
Are they bound to a bead?
Yes, they have some sort of a beads.
They
Yeah.
have a point?
Yes.
Okay.
Yeah.
And then for your B cell work, are you harvesting single B cell per well on the back end, or all of the, like multiple B cells that you assume are a single clone?
No, in fact, when we are doing a penning, some of the well ending up to have more than one cell. In that case, in those cases, particularly the way that we establish an assay, we tell the machine to go ahead and eject the extra one and make sure that each well has only one B cell. And then eventually when unpen them, we are going to have one B cell per 90 for the sequencing, basically.
One B cell per well,
Yes.
and that's what you grow your clones
Yes.
from that goes into the downstream assays.
Yeah,
as well.
normally we are lysing it right away
Mhm.
upon extraction from the chip,
Mhm.
and then making a cDNA and Sanger sequencing.
And the cDNA that you make on instrument that's bound to that bead, do you know anything? I mean, I understand that you're going to be sequencing VDJ essentially, right? Have you
Yes.
tried looking at other genes? I mean, have you tried looking at transcriptomes that are on those beads or anything like that?
No, we
No,
didn't
no.
do it. We got the instrument about 3 months ago, and we just got finished training, so we were able to run effectively after training only 2 assays.
Yeah. But that's
Yeah.
still a lot of progress
Yeah.
for a brand new instrument.
Yeah. But this instrument, one of the advantages, as I said, you don't need to only limit yourself to the B cell assay. It has a clear advantage if you would like to use the functional T cell assay. You can beautifully visualize it within a nanopen over there and monitor the toxicity that caused by your drug, or killing assay. You can perform the killing assay and lively track it within HPen.
Any other questions? If not, I'll just keep going. For your blooming assay,
Yes.
that looks fairly sort of explosive. Is it tightly tied to a pen? Are you confident you get the exactly right pen each time?
You will end up, that's depending, because machine work based on the microscopy basically. It depends how do you set up your assay, how many basically cycle of imaging you are requiring. For my assay, every 30 minutes is necessary, and after 30 minutes you can see a little bit bleed over, but you can always visualize the image that is very early in the blooming stage. So you do not facing cross-reactivity. Yeah.
Great. All right, and then, sorry, very last question. For when you, Simon, when you've got your what do you call them, flow cells, chips, and you're doing a long-term experiment in an incubator, you mentioned about changing media. Is that a delicate process, or is it fairly straightforward, like just routine? And I'm assuming you just do the whole thing off instrument, just like a regular TC situation. Oh, you can do it on instrument.
The instrument can do it. Usually, a lot of times people do it off instrument just to make it kind of that much faster.
Yeah.
Because then you can have one thing that's on instrument while you're exchanging media off instrument. You can do that kind of manually in a TC mode.
Yeah.
So, when it comes to caging, does it have to happen after you go in the cells, or you wait for a few and then caging? Like, if you're allowing, say, neurons to go for that?
Yeah. So we haven't done it, but Cellanome's R&D lab have done it. So they've allowed a lawn of neurons to grow. And I think I took the slide. It's on our website, actually, the MGC website. So allow the neurons to grow out to actually synapse, and they've actually tracked calcium signaling, not on the machine, but another scope that is capable of doing it. But you can cage on the cell bodies without crushing the axons. So you can allow the caging to happen after synapsing, growth and synapsing of neurons. So it looks very cool. Yeah.
No, I guess I was just trying to understand. I'm going to ask the question.
Okay. Well, thank you very much, everybody. Thanks for your time. And so we're ending right on time, actually. Great. Thank you.